regenerative properties, tissue repair peptides. And this is actually a spin-out from Lund University. It was founded in 2011, this company. We have two programs at the moment in our pipeline. The first one is FOL005, which is indicated for alopecia or hair loss. And this was founded by a serendipity. It was an investigation led by the founders of the company that, by coincidence, saw that this peptide, or actually the parent protein, could give rise to a strong hair growth in mice. And the other program is diabetes, a program which we hope to target both diabetes and its complications. So the FOL005 for alopecia is in clinical phase 2 at the moment, and the diabetes program is still in preclinical stage. So, as I said, the company was founded in 2011, and we are actually a public company listed in Spotlight stock market in Stockholm since November 2014. We are a virtual company. We're only about 5 to 6 people working here at the office in Lund, but we outsource most of our activities like animal studies or research, but we do collaborate a lot with the universities like Lund University or other universities as well. So let me first just take you through the FOL005 program. I just have a few slides about that. It's not an indication perhaps which is so common, and it's actually a high medical need for many people losing their hair, and that can be quite tragic, and problems with your social life, and especially if you are very young. And so we think that we can help people gaining their hair back with this treatment. So just a few words about science. So this is a slide. It's not very visible perhaps, but this is a section from skin where we have injected the FOL005 to female mice, and we have waited 20 minutes before we have taken out the skin and look for where the peptide actually is distributed to what kind of cells. And this is a strong, intense labeling of hair follicles. It's perhaps not clear in this picture, but it's really strong. It doesn't go to any other tissue in the skin. It's directed towards the hair follicles. And in this section, you see a human hair follicle. And in the middle of that structure, the hair fiber is coming out. And you can see the red staining is the peptide. It is bound to specific cells that we know control hair growth. So this is another way of showing that the peptide has a very specific function and binding to cells controlling hair growth. And this program is now in clinical phase 2. We have a clinical trial ongoing in Germany. And alopecia is a big problem, actually, for both males and females. Most often you think about males losing their hair, and it is indeed a very high need for products on the market. There are only 2 products currently on the market, Rogaine, which is indicated for both males and females. It's a topical solution, and you can buy it over the counter. And the other product is called Propecia. It's a tablet, only indicated for male patients because it's a prostatic compound from beginning. The market is quite big. It's over $3 billion globally today and increasing. And I think that has also a reason because we are so much aware of how we look. And if you don't have good hair growth, that could potentially be a big psychological problem. And for us, this is a key target, both males and females, because our product is very safe. We haven't so far seen any adverse events in our 3 clinical trials performed so far. And we hope that we can finally deliver a solution to this problem. So today, more than 90% of people that has problems with their hair growth is not being treated. And that could be due to the fact that these medications are not effective. They could be having adverse events, or simply that people are not compliant enough. They don't follow the treatment, because you have to be in treatment for more than 4 months before you can see a good effect of this treatment. So it's really a big problem. So the other program is the diabetes program. And this was actually founded a couple of years ago when we were studying mode of action of FOL005, because this was a serendipity finding. We didn't know to what cells it affected or the receptors. So when we studied the mode of action, we actually found out that these peptides, not FOL005, but other similar peptides, could actually play a role in diabetes. And potentially what we have seen so far is that these peptides are very potent in protecting beta cells from, for example, high glucose levels that could, in the long run, really depress the functionality of beta cells. And you all know that diabetes is a big market. Many people suffer from diabetes, and still, you know, despite of a lot of good products on the market, there are many people that remain untreated. Many products on the market, there are very old products and quite new ones as well. So as we are working with peptides, we have identified GLP-1 analogs as one of the, let's say, benchmarks to our program. So that's quite important for us to investigate how our peptides are performing compared to GLP-1s. And we are not only targeting blood glucose levels, we're also targeting diabetes complications. And you have a number of examples on the right side of the slide. And there are many, many different complications you can suffer from if you don't control the blood glucose levels or your diabetes. Some are very mild, but some are very, let's say, not so nice to have if you have to amputate your foot or leg. And there's also liver complications, which is not in this picture. So we hope that we can do a difference here. So how can a small company compete in like a very crowded diabetes world? So we work with Lund University Diabetes, LUDC, and this is a center in the university focusing on diabetes. And we are part in an industrial research collaboration where there are other companies also present in this effort to strive to bring new products to the market. You can see some of the companies on the lower left side. So the idea here is to bring forward new compounds, new strategies for combating diabetes. This gives us access to lots of resources, lots of models, lots of collaborations. And then we have an ongoing Eurostar grant, which we got quite recently with a few German partners, BioAssay and the German Diabetes Center in Düsseldorf. And this is focusing on the beta cell health. And this altogether is very valuable for us as a small company because we don't have anything in-house that can, let's say, compete with a whole worldwide network of researchers or partners. So just a flavor of what we have seen so far. So you see a mouse where we have injected subcutaneously on the back of the mouse a radiolabeled peptide. And you can see on the top of the mouse blackling underneath the skin. So that's where the peptide was located, in this case, 1 hour after administration. And you can see a great uptake in pancreas. So the peptide goes from the skin area to the pancreas. And the right panel, you see Langerhans islet in a mouse pancreas. Similar to the hair study, we have fluorescently labeled the peptide, and the red is the peptide bound to beta cells in the pancreas. So you can see here again, very distinct, strong binding to the target cells. And here is also just a taste of what we have seen on human islets. So these are islets coming from human donors, humans that has died but donated their pancreas to research. So on the left side, you can see an example with non-diabetic donors. And this is an experiment where you have exposed the Langerhans islets to high levels of glucose for a long time. And you can see if we add the full peptides, you can actually increase the insulin secretion. And we are on par with liraglutide. And the right-hand panel, you can see some examples of a type 2 donors, a similar setup. This is also a long-term incubation with high glucose. And you can see that the peptide actually performs quite well. So we hope that by improving or optimizing the peptides and also studying other complications related to diabetes, hopefully we can offer an interesting product to bigger pharmaceutical companies, because as a small company, we cannot bring this further into clinical development, maybe clinical phase 1 or phase 2A, but we can't bring it further. So that's a short presentation of Follicum in a nutshell. I will just try to switch to I need to go back. Okay, so this is my sort of personal reflections on my career in life science. So I'm almost 60 years old. I've been working in pharmaceutical industry for a long time, maybe too long, but it's a very interesting field. So I live on the coastline, just outside or north of Malmö. I have 2 dogs, 2 boys, and a wife. You perhaps joke about the order of it, but it's my life. My interests are woodworking, renovating houses, dogs, hunting, golfing, food, and travel. So I try to mix this with my let's say industry life, because I think it's very important to have a life outside what you are doing, and research can sometimes be very, very time consuming. So by this I get my energy. I enjoy being out in the nature, walking and thinking, because in the back of my head I can then solve problems on what I have been doing in my work, or if the problems are quite severe, it helps me out really much to play around with the dogs, train them, and everything like that. So my background is I have been studying in Lund University. My background is more or less pharmacology from training. I did my Master of Science in Lund, but also the PhD at the Pharmacological Institution, and the Medical Institution in Lund. And I mixed this education with a lot of practical work on the lab. And after being finished with my academic career, I went to big pharma and then subsequently to medium, and I had a couple of years on a food tech company, and then I went back to small biotech where I am now. So I started out in Pharmacia, which was a big Swedish company. It's now part of Pfizer. I started out there with a research program in oncology. So I was working with, you could say, particular kind of compounds indicated for prostatic cancer. So I was heading out a program for investigating whether certain, you can say, class of compounds could affect prostatic cancer, because Pharmacia had that as one key indication. And in that time, Pharmacia was bought by another company. It was merged. It became Pharmacia Upjohn. So Upjohn was quite a big company in the US. And at that time, when the merger was finished, the big company was changed quite a lot. And my project moved to Italy, because they had a site in Italy. And then I was starting to work with clinical trials instead. So I went from the lab bench to working with clinical trials, oncology trials, and also some immunology trials, which was quite interesting. But eventually, the company was becoming too big, and I was feeling a little bit, too narrow. I enjoy working not to, let's say, to be too specialized. So then I moved to Ferring. I was part of a program, and this was the first time I was working with peptides. And this was in the end of 90s. We had a big phase 3 program at that time. So I was responsible for finalizing the clinical phase 3, writing part of the new drug application to the EMEA. And in the end, we got this approved. So actually, very good experience to get a new product approved by the centralized procedure in Europe. We ended up with a hearing in London, and finally we could get this approved. So it's now actually on the market. The indication was preterm labor, so we had both the mother and the baby as patients, and the idea was to reduce the contractions on the mother's side, so the baby could stay healthy inside, or reduce the time, let's say, so you could give some medications to improve the survival of the baby if it was delivered, let's say, week 24 or 26 or 28. But it's a successful product on the market. And at that time, I worked both in Sweden and Denmark. And then from that, I went on to a small, you could say, biotech company, Bioglan. It was a UK-based company. And part of it is still here in Malmö. It's a company now that is a very small part of what it was from the beginning. It's a company that had, at that time, a lot of dermatological products on the market in Europe, but also research programs on drug delivery, both parenteral and topical programs. So I was headed the clinical and regulatory department on Bioglan at that time. So I got insight in how products on the market works when you have to take care of safety issues or periodic safety updates and all that. And then I moved to Sky Pharma and followed some drug development on Sky Pharma, mostly on parenteral drug development, just a few years, and then that was moved to San Diego, but I didn't want to move. So I started to work on probiotics. Probi is a small food company based in Lund, and they work with lactobacilli in product you can drink or in food supplements like capsules or tablets. I had a few years of very interesting work. I was the CSO of that company. It's quite interesting. It's quite different to work with food companies compared to biotech or pharma companies, but you still do clinical trials with food companies, and especially if you want to have health claims on the products on the label. And then after that, I moved to a small company here in Lund. It's also based on peptides, Dermagen. It was antimicrobial peptides coming from ourselves, from the complement system. And then it was merged to a little bit bigger company called Follicum. And finally, in 2013, I was employed by Follicum. And now we are back again, working a lot with academic because we are outsourcing a lot of activities to different academic groups. And you saw again in our diabetes program that we have a lot of interactions with different academic groups. And for me, that is very exciting. You can, you have the company work, but also have the academic work. So this is actually in between, a very nice way of closing the circle, experience from big pharma, and then going back to small biotech. And that's quite nice. So reflecting a little bit on my roles and positions in the industry, I've been working in different companies, different positions, different, let's say, roles. And at the time, if I look back, I didn't have any specific goals for reaching out a certain position or a certain company. And that was more, let's say, coincidence, really. I'm very goal-oriented. I think it's really good to be that, and also a team player, because working in small or big companies, you need to be goal-oriented and a team player. But again, realizing that, coming from academia and then moving into a company, depending on the position, of course, but you move from being a specialist to a generalist. Today, I'm working with a lot of different things, from approving invoices or purchasing to financing to research programs. So it's a menu that is quite big. And if you don't like that, then maybe perhaps the position in a small company is not of interest, and maybe a position in a bigger company is much better. And I guess, yes.
And there is a question from Marie.
Okay. Yeah.
She asked, did you apply for the different jobs? Did you use your network, or were you recruited, or perhaps a mixture?
Yeah, it was a mixture. It was the first jobs I certainly applied for, but then it was a mixture. And suddenly when I was, when I saw, for example, working in a bigger company, this was in the end for me quite boring, and I felt, I couldn't give 100% because I thought that the company was not doing the right thing. Then I moved on, applied for a job at Faring, for example. But then after that, more or less, the other jobs were I didn't apply for. I was recruited, more or less. Yeah. So today, a normal workday in my life is to consider this. You all know this funnel. You have ideas and you try to make them as projects. Early projects, maybe not suitable for pharmaceutical industry, or driving into the funnel, then they are coming outside. But eventually you find a project you would like to pursue into a discovery phase and then preclinical stage. And before going into clinical development, you need to do safety tox investigations. And this is the thing that we think about all our time, more or less, for our projects, irrespective if the project is in the early stages or in the late stages. We need to think about how to generate value, because we are funded by investigators, investors, and they would like, of course, to have value for the money, and of course, also in the end, a good yield of their investment. So this is traditional. This is not unique for biotech. This is the same for big pharma. But for a biotech company, fewer persons need to think about this, and it can be quite complex. You need to think about, let's say, research. How do you do the best research to push the program further into the funnel? How do you take care of, let's say, intellectual property? How do you secure that you are secret? Because if you, as us, are listed on a stock exchange, you can't disclose any research to the market if you don't do it by press releases, to make sure that everybody get a chance to see what you're doing at the same time. Of course, you can work around to have non-disclosure agreements and such things, but it's really important that you keep things in-house. Other things is, of course, that the indication or the project you're working with is or having a medical need, or is possible to develop. A lot of ideas here will fail because you don't see a market, you don't see a possible straight way through the funnel, and perhaps there will not be any investors to support your development. So this is something that is a headache sometimes. And I guess when I worked in the academia, most of my funds came from grants. But in small biotech, you have external investors. And I think in a big company, you have sales that contributes to what you're doing. But in a biotech company like ours, we don't have any sales. So we are funded by doing share emissions and asking investors to believe in us. So we have to present what we are doing in a very confident way, and really to attract interest from investors. And initially that can be quite hard. And perhaps starting a small company, you have fools, friends and family that invest in the company, but then subsequently you need to bring in more money, and you have to have bigger investors, and that doesn't disclose or prevent you from applying for grants, like we have done with the EU grants, or through collaboration with the university, we can apply for grants, and we let the university use our peptides, for example. So it's a mixture, and in a biotech, this is also a major headache, and is very much part of my normal workday, to think about keeping the investors happy. And of course, to generate value in a small biotech, there are lots of challenges, and you have a list of a few here. But again, to be really, really honest and frank, you have to prioritize very, very hardly, and you have to keep focus 100%, because if you don't, the valuation and the visibility of the company will go down. Your competitors will perhaps be more efficient in the development or research, or even grab your resources. So financing the company is very important, and by doing so, you have to be very attractive. So you have to select the right indications. And again, to create a small biotech, you need an organization. You can have it as ours, virtual, or you can have it a little bit bigger. And you could be completely virtual by just working with consultants or contract research organizations. But in the end, to really go through this funnel, you need a partner. And that is something you need to keep in mind from the start. Who will be your licensing partner or the partner to collaborate with in the end? Who will be the one that buys this or take over your product development? And if you are a funder, this is your baby, this is something that you need to consider early on, not to be, let's say, stuck too hard with the project or the company. And again, looking at the funnel in another way, there are, of course, very many gates where the products or the projects can fail. You have to have a continuous surveillance of the performance of your products. Are they still of interest externally to investors, still medical need? And you know that many projects will fail during development. So it's a constant filling of new products in this development, which is also part of my daily work. And this is my last slide, so I hope I have shown you some challenges, but also I hope you've been interested in working in a biotech, because it is really important that we start new biotechs, because the big pharma, they cannot fulfill the need of biotech companies, but they can definitely buy biotech companies and the projects to fill their pipeline. Because in our world, we hope that we can find by the end of the rainbow a big sack full of money to satisfy our investors. So the funding is very important, but also that you have the right competences and experience in the team to really drive this forward, and also to understand where your limitations are, because resources are very much scarce and it costs a lot. And again, then you have to decide on how long in the product life you're going to take it, if it's only preclinical or it's a clinical program or clinical phase 2 or phase 3. So you have to decide that early on, and together with your investors, of course. But it's really fun and interesting. It's very much different from a big pharma. It's much more, let's say, individually driven. And I think I would recommend to all of you that are interested in this to have a second thought, maybe to start out working in a bigger pharma, but then definitely go to biotech company, because it's more rewarding, I think. So thank you very much for your time, and any questions are very much welcome.
Yes, there is a question from Payot. I think it's pronounced. What kind of IT skills, mostly focusing on computational biology, data science, research, data management, if any, do small biotech companies like yours need?
Not sure if I understand the question. So we don't do that in-house. We outsource that to specialized companies.
Okay, maybe you could unmute yourself and then elaborate a bit more if you didn't get an answer to your question.
Sure. Hi, Jan. Thank you for your presentation. That's sort of exactly what I'm asking. So, because I do realize that probably you don't have any such need, or these needs are not pronounced enough to have in-house specialists dealing with this data. But
that's right. Yeah.
But I would assume that you're working with external partners, as you've mentioned. And
yeah, absolutely. We are working with. Yeah. Sorry. Please go ahead.
Could you describe briefly what kind of skills you need, or what kind of needs do you have in that area, and what would you be looking in partners?
Can you, so what kind of skills are you thinking of? Is it computational skills for bioinformatics, or is it more like, clinical trial data sets, or is it more optimizing, for example, chemical structures of our compounds?
So maybe let's focus on the bioinformatics and then the management of clinical trial data.
Yeah. Okay. So bioinformatics, we are working closely together with the University of Lund, for example. So LUDC in Malmö, they have lots of bioinformatics specialized people that work with that. I'm definitely not an expert, but if we have questions, we go there first. If they can't help us out, we go to specialized companies and try to collaborate with them. So this is typically what we do. This is not something that we do on a regular basis. It might be, let's say, we have even this need maybe 1 or 2 times a year. For clinical trials, we do that with a partner that take responsibility of the performance of the clinical trial, because that's very complex. So typically what we do, we go to a CRO specialized in clinical trials, and we say that, okay, for example, in our hair study, we would like to do a hair study. We would like to do it in Germany. And before doing so, we have a very clear view of what we would like to do. So we have typically selected the clinical sites before we go to this CRO, and we have typically also a good idea of what kind of study we would like to do. And then they take care of everything after that. We are not involved in the databases or the collection of the data, but we are involved in the analysis of the data. Together with the CRO, we are setting up a statistical analysis plan where we have the endpoints clearly defined and how we would handle the data in the end and so on.
I see. Thank you so much. That exactly answers my question. Thank you.
All right. Thank you.
And there's also a question from Jonas. He asked, can you elaborate on the pros and cons on working in 2 such different fields as hair growth and beta cell function?
Yeah, that's also a typical question we get from investors. What do you know what you are doing? But basically, the scientific part is more or less the same. Actually, the hair follicle needs insulin to be able to grow and generate a good, strong hair fiber. So there are several connections, both biological ones, but also other ones. But basically, the research, and I would perhaps say the development of this program is the same. You have guidelines to follow. So once when you have selected a molecule and you are moving towards clinical trial, you have to fulfill the regulatory guidelines. That is, they are issued from the authorities like the EMA or FDA. And these guidelines you have to follow for all medical or pharmaceutical products. So it's not that different. But then again, you have to involve expertise. And of course, the expertise in the hair, they are much different from the expertise we have in the diabetes project. So what we have done is we have set up scientific advisory boards, one specific for the hair project and the other one is specific for the diabetes project. So that's how we do it. We are not experts of both, but we over time we learn a little bit. But you can utilize lots of what you have done, for example, in the hair project, and that can be applied also in diabetes or any other project.
Okay, thank you. And there's a question from Peter. He asked, thank you for your inspirational presentation. I'd like to know how much your company is open to hiring medical advisors with a clinical background.
Yeah, so as I said, we are trying to keep the organization as small as possible, and we are doing so by having a small team, but we are working a lot with consultants. So we typically don't hire people. Basically what we do, we work with consultants, and this is the way we do it. And the reason is that it's quite costly to recruit a person. And for example, a person that, or a position that is only needed, yeah, let's say 20%. It's difficult to recruit one person that is only working 20% for us. Typically, they would like to work full time, and therefore we work most often with consultants. Sometimes we can have part-time employees, but it's not the best for us.
Okay, thank you. I think that was the last question for now, but you of course still have